All right, let's kick in meiosis. Right, meiosis is different from a division of a cell that you did in grade 10, which is called mitosis. Remember, in mitosis, one cell divided into two identical cells, genetically identical, right? And then if the cell started off with 46 chromosomes, for example, the two cells will end up with 46 chromosomes each due to DNA replication. So that was mitosis. Now, meiosis is different. It is not that. We'll go into it and you'll actually see that it's very different from mitosis, right? Good. But first things first, what do you need to know? What do you need to know before going into meiosis? You need to revise your cell structures, your organelles, for example, especially the nucleus, right? Because that's where meiosis basically, majority of it, we're going to look into the nucleus as to what happens. Then you need to revise what are centrosomes and the cytoplasm and so forth. Yeah, good. Some of the things that you need to know is that you need to know that each human cell will consist of your DNA. And what is a replicated DNA? What is an unreplicated DNA? DNA basically consists of that DNA and proteins which make up your chromosomes, for example. So which proteins are we talking about? We dealt with this when we were doing replication. Those are your histones, for example. Your histones and DNA together coiled up, they make your chromosomes. Good. All chromosomes in a human somatic cell are 46, right? Good. And then they can replicate during DNA replication and make a copy of themselves. Great. Now, also, a certain terminologies that you need to be aware of is haploid, for example. What is a haploid cell? What is a diploid cell? Then you'll need to know what are gametes. Gametes are sex cells, basically your sperm cell and the ova, right? We don't like calling it an egg. It's a bit confusing when we do a reproduction. It's not an amniotic egg, it's an ova. Yeah? Good. So just as we call the male one a sperm, the female one is it's an ova. Then somatic cells, those are all your body cells except for your sperm or the female ovum if you're a female. Then sex chromosomes. Sex chromosomes include your gonosomes and autosomes, which we'll look at right now. Then revise mitosis. Good. Now, meiosis is a continuous process, but in this case, we're going to divide it into two processes, right? Good. That is meiosis 1 and meiosis 2. Everything starts with what? Everything will start at interphase, where DNA replication takes place. Good. Also, what do you need to know? You need to know what a karyotype is. Ne? You need to know what is a karyotype. A karyotype is basically a representation of the shape, the arrangement, the number of a full set of chromosomes in the nucleus of a somatic cell. Emphasis of being on a somatic cell. Good. So how those chromosomes are arranged, their shapes. So they are arranged according to their shapes. And then the number of those chromosomes. So basically they are arranged in pairs. A total of your karyotype is 23 homologous pairs, but the total for all of your chromosomes is basically 46. They exist in pairs in a karyotype. Good. Up until when? Up until the 23rd one. Then you need to know the difference between a diploid cell basically in a human being. In a human somatic cell, it will consist of 46 chromosomes. A haploid cell will consist of half the number of chromosomes, basically your 23 chromosomes. Not 23 pairs, but 23 chromosomes. You need to be careful of that. Good. In your karyotype, we have what we call autosomes. All of those chromosomes in your cell, in the nucleus of your cell, that do not determine your sex, basically, or your gender. Basically, they do not determine whether you're a female or you're male. Those are called autosomes. Those chromosomes are called autosomes. And the ones that determine your sex or your gender, basically are called your gonosomes. Ever heard of the XX and XY chromosomes? That those are the 23rd, that is the 23rd homologous pair of your chromosomes that determines your sex, basically. And then you'll see that they exist in pairs, as I've already said, in what we call homologous pairs. A pair of matching chromosomes in an organism, with one being inherited from each parent. Remember, you get 23 pairs, 23 chromosomes, not pairs, from your father, and 23 chromosomes from your mother, making it 46 chromosomes all in all, and they will exist in pairs. That's how it's represented as your karyotype right over here. Good. Let me move. Good. So, as I've said, in interphase, just to summarize the whole entire meiosis. Meiosis will start in interphase, then meiosis 1, and then meiosis 2. What happens in interphase? In interphase, you'll have chromosomes. For example, in this cell, I have 2 chromosomes, right? And those 2 chromosomes, during interphase, they are going to undergo a full DNA replication. And once they replicate, they will replicate, and then we'll end up with 2 replicated chromosomes. So that's the difference. This is an unreplicated chromosome. This is a replicated chromosome. How do you see that as a replicated chromosomes? It has chromatids. So once they are replicated, they are called chromatids. So to give you an example, how many chromosomes do you see in cell A? 1, 2. You count the number of chromosomes by the number of their centromeres. That's the central structure in each chromosome. Then, in the second cell, cell B, how many chromosomes do you see? Still 2 chromosomes. How many sister chromatids are there now? There are 4. Well, we don't call them sister chromatids anymore, but how many chromatids do you see? 4 chromatids. These are not chromatids, they are chromosomes. They can only be chromatids once they replicate. So please get that right. So after they replicate, they will undergo meiosis. Those replicated chromosomes now will homologous. They are homologous chromosomes. They are separated. The blue one, let's say for example, the blue one is from your father, and the red one is from your mother. So they will separate into different cells. And then we have the number of chromosomes. Now, those cells by the end of meiosis 1 are haploid, but we're having duplicated chromosomes. And then those duplicated chromosomes now are in each cell. In meiosis 2, it has to do with now with the chromatids. The chromatids, let me cancel this one out. We don't use it in caps. Those chromatids are now separated into 2 cells, meaning they are still haploid cells. At the end of meiosis 2, we're going to have 4 cells that are genetically different and are haploid. Good. With that being said, let's get into meiosis. So with meiosis, I like to draw. Let's start with interphase. So during interphase of meiosis, the main thing that happens is what? DNA replication. DNA replication. Obviously, your DNA will be in the form of chromatin network. It won't be the typical DNA that a typical chromosomes. It will be in the form of chromatin network. But in this case, I just want to make it a chromosome so that you understand what I'm talking about. If we start off with, let's say this is DNA 1, and then its homologous pair is this one. Then another homologous pair over here. How many chromosomes do we have in this cell? We're starting off with 1, 2, 3, 4 chromosomes. 4 chromosomes. Now, during interphase, DNA replication will take place, whereby they make a copy of themselves, all the homologous pairs. So this one makes a copy of itself, the one from your mother, and the one from your father makes a copy of itself as well. Similarly, the other homologous pair does the same thing. This one from your father, your mother makes a copy of itself, and the one from your father makes a copy of itself. How many chromosomes do we still have? We count the number of centromeres. We still have 4 chromosomes. Now, how many sister chromatids do we have? We have, or how many chromatids do we have? Not sister. We have 1, 2, 3, 4, 5, 6, 7, 8 chromatids in the cell after replication. Good. Now this DNA replication is taking place. Now this cell is ready for meiosis, for that reduction division. Good. Let's go into meiosis. Remember I said meiosis is divided into 2 parts, meiosis 1 and meiosis 2, both of them consisting of the phases of meiosis similar to mitosis, prophase, metaphase, anaphase, telophase with cytokinesis. So let's look at meiosis 1. Meiosis 1. Meiosis 1 will start with prophase 1. And prophase 1 is the most eventful, I'd rather say, it's the most eventful phase and one of the most important ones of them all. So what happens during prophase 1? We're going to take those chromosomes that we had. Those chromosomes, CS1, it's a replicated chromosomes in a chromosome, remember? Those homologous ones. And then the other ones, slightly taller. And then its homologous partner over there. Now, what happens during prophase? Similar to mitosis. During prophase, the nuclear membrane and the nucleolus start to disappear. And then the two centrosomes begin to form spindle fibers and move to opposite ends of the cell. So this one will move here and this one will move here. So that is your centrosome. So the centrosome and then the nuclear membrane disappears. Also the nucleolus disappears. But we're not really interested in that. No. What's more interesting now, during prophase 1. Prophase 1 is all about genetic variants. We need to recombine the genes now. In prophase 1, an important process takes place known as crossing over. First things first, these chromosomes will line up, the homologous chromosomes line up very close to each other. They line up close to each other, forming what we call a bivalent. Now, after they form those bivalents, they are getting ready for crossing over. What happens in crossing over? Now, let's draw them down here to see what happens in crossing over. We have this one and its homologous partner. And then we have that other one with his homologous partner. These are the bivalents. Now, the inner chromatids, will overlap during crossing over. What do I mean? Look, there's the outer, slightly shorter. This is the outer chromatid, centromere, and then the inner chromatid. Similarly, the homologous partner, outer chromatid, and the inner chromatid. So the two inner chromatids, these ones, they overlap. When the two inner chromatids overlap, the place where they overlap, that they form what we call a chiasma. Where they overlap, they form what we call a chiasma. Similarly, the other ones as well, the other homologous pair will do the exact same thing. Remember, that's slightly taller. partner overlaps, centromere, also forming a chiasma. Many are called chiasmata, one is a chiasma. Then what happens? At that point, they are gonna exchange genetic material in such a way that we are gonna have this one from your father having a bit of DNA from your mother, and then the DNA from your mother will contain a bit of DNA from, whoa, no battery, will contain a bit of DNA from your father. So they have exchanged DNA. That is during prophase I. Also, the other homologous pair does the same thing. It will end up with DNA from your father with a bit of DNA from your mother. This is an exchange of DNA. Then your mother's DNA has a bit of your father's DNA. Remember, these ones are slightly longer. Let's make them slightly longer. They will look something like this. Great. That's after crossing over. Now we have a recombination of DNA over here. Now we have genetic variation in this cell. So this process takes place where? During late prophase. Early prophase, what happens? All of those centrosomes move apart, spindle fibers are formed. They will connect to the centromere of each chromosome, and then your nucleolus disappears, nuclear membrane disappears, bivalent is formed, and then during late prophase, that's when crossing over happens, the exchange of DNA. After this happens now, let's count. This is gonna be in your, let's do it like this. All of this is gonna be in late prophase I. This is still prophase I. Prophase I, in that cell during prophase I. We are gonna end up with this. This is what we call late prophase I. So still prophase I. Now we have this. Now, what else are we gonna have? We're gonna have those centrosomes at different poles of the cell, forming spindle fibers. Now the spindle fibers are going to attach to what? They are going to end up attaching to each chromosome, to the centrosome, the centromere of each chromosome. And then these chromosomes now are going to start moving to the middle of the cell, the equatorial region of the cell, in preparation for what? In preparation for metaphase. So let's quickly go back to this. During the centrome splits and the two centrioles move apart. Then from the bases into individual homologs like each other, forming. Then in crossing over takes place. What is your inner parts of homologous chromosomes overlap with each other, and then they exchange DNA material, or they basically are going to play, and they do this with such precision that it does not interfere with the coding of any gene or protein in your body. That's brilliant. So what is the main purpose of crossing over in prophase I? Is for crossing over is for basically genetic variation. It brings about genetic variation. Good. And that ends our lesson on interphase and prophase I. We'll continue with the other phases in the next lesson.